Welcome to my YouTube channel. Farmers' profits are decreasing day by day due to the several reasons like increase in cost of inputs like fertilizers, pesticides, weedicides, etc. And there is an increase in labor cost also, and there is leading to the decrease in crop production. All these reasons leads to the profit reduction, even though the procurement price of the crops government is increasing year by year. Due to all these reasons, there is a need to reduce the cost of production, to concentrate in precision agriculture and to apply inputs wherever and whenever it is required. We have to concentrate on the use the inputs wherever it is required and whatever it is required. So concentrating on this, we can reduce the input cost, and also there is a need to use the IC tools and digitalization of the agriculture. All these are needed to decrease the crop production cost and increase the farmers' income by double in coming years. So, keeping in view the importance of this precision agriculture technology to boost up the crop farming, this lecture is being taken to address the students, farmers, and scientists and extension workers. Precision agriculture systems are dynamically developing management systems. They allow farmers to solve a whole spectrum of problems. At the same time, precision agriculture includes a wide range of tools that growers need to master to get the most out of their production. Among the technologies used in such a system are, for example, geoposition system, GPS, drones, and satellite images. Based on this data, farmers receive information on all key issues like crop status, weather forecast, environmental changes, etc. Also, the important difference between the precision farming and traditional agriculture is the ability to manage fields not as a single block but by dividing them into separate areas. Such zoning allows diversifying the management decisions for individual field plots, adjusting the fertilizer amount, optimizing the techniques movement, and using fuel more frugally. The total plat, it may not be having same nutritional nutrient status. It may be having different characters, soil characters, and different nutrient status. So we have to divide into the field in the small units. Based on the status in that small unit, we have to concentrate on the inputs application just like fertilizers, so that we can increase the yield and put down the cost of production. Precision agriculture systems are dynamically developing management system. They allow farmers to solve a whole spectrum of problems. At the same time, precision agriculture includes a wide range of tools that growers need to master to get the most out of their production. Farmers can control the process remotely with a precision agriculture system. Even small farms can manage large fields or a group of small areas. In dramatically, it improves the efficiency of the crops and saves the financial costs while increasing the production. The last aspect is essential since it seems that precision agriculture technologies are costly at first glance. However, the savings are significantly higher than with the traditional agriculture methods in the long run. So, growers can accurately sum the required fertilizer amount, determine the effective fertilizer types for a particular area. So, it is a long run, it will lead to profits and it may be useful. So moreover, the importance of precision farming technologies is that they improve the planning of agricultural operations for an extended period, adjusting the real-time strategy during the force. Optimizing the soil use preserves its quality, enabling a stable food supply. Therefore, the precision farming in agriculture plays an essential part in solving the global problem of hunger. What are the benefits of the precision agriculture? There are several benefits. Precision agriculture is helpful for farmers or growers and the environment as well. Moreover, these areas are interconnected since environmental degradation worsens conditions for agriculture. Here are just several benefits of this precision agriculture. Number one, most important is minimizing the cost of materials, inputs, and resources like water, seed, fuel, etc. Maintaining the soil health by reducing the number of pesticides and excess use of the fertilizers, lowering the agricultural dependence on weather conditions, maximizing the realization of the genetic potential of the producer crops. All these advantages of precision farming allow farmers to improve the quality of products significantly and at the same time it reduces their costs. Precision agricultural technologies, it requires special equipment and software to collect and analyze all the information available. Since precision agriculture technology uses hardware and software also, specialists are needed to implement them. However, with further work, farmers can cope up on their own. The precision farming technology can be conditionally divided into ground, aerial, and satellite. The former is suitable for production planning, mapping, scouting, and machine control. The last two one, that is scouting and machine control, are relevant to the solving more global problems. For example, real-time yield state analysis from anywhere. It is recommended to mix the technologies to obtain the objective data. The global position system, that is GPS, the role in precision agriculture. The GPS refers to ground-based technology permitting growers to gather or to collect the data with accurate location and information in real time. So GPS is suitable for the different tasks like mapping of irrigation systems, fields, and roads, and detection of areas with problem plants, soil testing in specific field areas, the tractor driving with a parallel steering system, VRA for precision seed and fertilizer application, that is the VRA means that is variable rate of application. And also the GPS in precision agriculture allows for controlling agricultural machinery. For example, with the help, growers can drive tractors in poor visibility due to rain or fog. The GIS technology, in the role of GIS technology in precision agriculture, that is, the GIS is nothing but geographic information system. It operates with object details and location data to create maps, including digital ones. For example, for dividing the farm into separate zones, remote sensing is needed. All zones have their characteristics, the analysis of which requires GIS and GPS use. Traditionally, these zones are divided according to soil type, nutrient availability, soil moisture content, pH rate, and pest infestation. So the geographic information system in precision agriculture permits farmers to view records such as soil survey maps and plant characteristics traditionally grown in the region. Satellite images and aerial photographs provide additional information. Another handy feature of GIS is analyzing multiple farm management options by comparing and manipulating the data layers. The UAVs and dusters in precision agriculture. Aerial technologies for crop management are based on using unmanned aerial vehicles. The UAV means unmanned aerial vehicles. Thus, farmers can monitor the yields condition without scouting all fields in person. At first glance, the easiest option is crop dusters because farmers already use them for watering and sowing. They can attach a hyperspectral camera to any agricultural aircraft and record the needed information. However, it will lead to an additional load on crop dusters, which will be involved in addition to the main tasks for crop management. It will lead to faster wear of the devices and additional environmental pollution. In addition, this method means the employment of a qualified pilot. So nowadays, the farmers, progressive farmers, and latest technology, they are using drones for identifying the pests and for identifying the moisture status, for identifying the soil nutrient status. So like that, we can attach one camera so that we can survey the areas and we can map the areas. So using unmanned aerial vehicles or drones in precision agriculture is a more progressive solution. They are controlled remotely and consume less fuel. Moreover, the precision agriculture drones can analyze the field thoroughly, conducting complex multispectral thermal and hypospectral soil analysis. Satellite remote sensing in precision agriculture. This technology allows growers to observe the yield health using satellite images. They provide up to date information on moisture stage, disease, structural anomalies, and the nutrient levels. Modern precision agriculture satellite imagery has a high spectral resolution, allowing growers to get the most accurate data. It is most effective to combine scouting with satellite remote sensing. Growers can determine the cause of deviations from the norm. Also, they can use satellite imagery for planning plant treatments and selecting the agricultural chemicals. An essential benefit of this method compared to the GPS and UAVs, that is unmanned aerial vehicles, it is most benefit of this is the absence of the additional fuel and labor costs. How will use this precision agriculture and its application in the field? The agriculture system constantly evolves, permitting growers to optimize the production and exploitation of the resources. Therefore, farmers can improve soil quality and productivity. Here are the most popular application fields. So, VRT in precision agriculture, that is various variable rate technology. This is VRT, nothing but variable rate technology. The variable rate technology allows growers to apply fertilizers, chemicals, seeds, etc. to different parts of the field depending upon their needs. Also, farmers can test the soil for nutrients such as nitrogen and feed only those areas that lack of certain nutrients. Wherever the deficiency, wherever the lacking of that particular nutrients are there, there only we can feed the that required or deficient nutrients. So the variable application will be there. With that, we can reduce the cost on the inputs. So with the variable rate application, precision agriculture requires a differential global position system, that is DGPS, and specialized software. There are several variable rate technologies types are there. The map-based VRT corrects the number of applied fertilizers, pesticides, and other products according to the previously generated area map. The sensor-based VRT examines the soil with sensors in real time and can help determine the nitrogen deficiency. Then the control system estimates the required number of inputs. As a precision agriculture software, that is EOSDA, that is nothing but that is EOS data analysis technique. As a precision agriculture software, this is one software, EOSDA, crop monitoring. It provides the ability to zone any field based on vegetation indices. A zoned field map can be integrated with the variable rate technology to vary the amount of water, fertilizer, seeds, or chemicals on different parts of the field. So, thanks to the variable rate technology, farmers can care about plants more effectively, use as many additional resources as needed, save their money, and positively impact the environment. There is no fertilizer runoff. Is EOS data analytics, that is EOSDA. It manages the agricultural risk and receives timely notifications. So the software is very much useful in monitoring the fields remotely, get insights and make smart decisions all in on one platform, identify the problems and risks in the fields to make effective decisions, simplifies the simplify and speed up the precision crop analysis and vegetation health monitoring. It optimizes the resources and farm operations with satellite-driven features. It gets the comprehensive crop analysis. It monitors the crop health remotely with satellite images, and it gets notified about the risks. It optimizes the resource usage and customizes the reports. Field monitoring and mapping in precision agriculture. When creating a field map, it is necessary to consider the environmental condition to obtain objective data, such as landscape, earth, and weather. Therefore, the GPS and satellite remote sensing are used for this. So thus, the maps display all the field enhances and harvest data. Based on such precision agriculture yield map, USDA crop monitoring functionally allows growers to effectively monitor the readiness of the crops for harvest with the help of vegetation indices. So thus the NDRE index, that is NDRE is nothing but this is normalized difference red edge index. Normalized difference red edge index, that is NDRE. This index is excellent for such purposes, which more clearly shows the state of vegetation in the latter stages of the culture's development. It has to analyze the vegetation along with the entire depth of foliage, not just analyze the surface as NDVI does. That is NDVI means normalized difference vegetation index. Normalized difference vegetation index. Then historical weather and field data allows farmers to predict the crop yields. At the same time, such data must be accurate. With the precision farming, this API solutions by USDA crop monitoring, you can get the custom solutions on the yield prediction. And then coming to micro irrigation, that is nothing but drip irrigation. A micro irrigation system allows growers to effectively plan the field's irrigation by identifying the areas with high or low soil moisture. Precision agriculture irrigation makes it possible to carry out the variable rate irrigation. That means wherever the moisture is less, wherever it requires more irrigation, we have to give. Wherever moisture is high, it requires less irrigation, so we'll give the irrigation water, less water as irrigation. So that is the variable rate irrigation. So it is possible to carry out the variable rate irrigation based on the soil moisture levels to vary the water supply volume for different field plots. This level of control can significantly improve the irrigation efficiency and result in significant water savings. One of the indexes used in USDA software crop monitoring is NDMI. NDMI is nothing but National Disaster Management Information System. This index shows the crop water stress level in the selected field. With it, farmers can quickly identify the areas of the field that need additional watering, regions of flooding, and areas with excessive moisture. Site-specific crop management, it is very important. It is an agricultural management concept based on the monitoring, counting, and reaching to crop variability between the fields or within the one area. So this is nothing but in the same field, one plat, large plat, there are several variations within the plat, one corner to another corner, and at different places within the same plant. So the variable soil fertility status may be there, variable soil conditions may be there. So based on the nutrition status and based on the moisture status, based on the soil type, that that is nothing but spatial variability. Depend upon the variability within the plant, we have to plan applying the fertilizers, pesticides, whatever it may be. So that is the site-specific crop management. Most current site-specific crop management methods use accurate global positioning combined with the site-specific measurements to quantify spatial changing field conditions. They are field data collection such as pest presence and remote sensing data such as from satellites. So USDA crop monitoring allows growers to use management zones in precision agriculture effectively. It can identify the problem areas in a field with various possible pathogens, pests, fungus, fertilizer misuse, weeds, lack of moisture, etc. Once identifying a site with deviations, the farmer can send a scout to make a report based on the data collected during the inspection. So how we'll prepare the soil mapping in precision agriculture? So the precision agriculture is possible without quality ground mapping. With the help, farmers evaluate the soil properties, its chemical composition, the nutrients presence, etc. Soil mapping practice as existed for a long time, but modern technologies provide even more detailed information, making the new generation of digital maps more efficient. For obtaining the data, farmers use several types of precision agricultural sensors like optical resources, which interpret data based on the coefficient of reflection of light from the ground. Another one, electrochemical sensors, which analyze the soil's electrical characteristics such as the potassium's presence, for example. So another one, mechanical sensors. It in contact with the ground determine the types and density of the elements contained in it. So these are the different types of sensors in the precision agriculture used. Internet of Things used in the precision agriculture, this system gives farmers more control over the field with the dedicated data sensors, remote control, and IoT platform. With IoT-based precision agriculture, growers can control all the most critical information from air temperature to soil conditions. Moreover, technology solves the problem of manually researching large firms by collecting data independently. Also, robotic systems are increasingly being introduced which take on the part of a person's work. For example, innovative irrigation technologies free up human labor for other tasks and use water more rationally. The use of artificial intelligence in machine learning in agriculture, coming to this aspect, the artificial intelligence and precision agriculture are constantly being improved and provide new intelligent tools for managing agricultural production. Image-based pattern recognized systems allow applications to adjust the watering and nutrition of the plants according to their type and requirement. For example, sprayers equipped with artificial intelligence can recognize the specific weeds and spray them without affecting the crops being grown. So there are also some challenges in precision agriculture. The goal of sustainable agriculture is to meet the current needs of the society, primarily in food, without endangering the environment. Precision agriculture conserves resources by allowing farmers to cultivate crops and soil with maximum efficiency. Artificial intelligence and machine agriculture are constantly improved and provide new intelligent tools for managing agricultural production. Image-based pattern recognized systems allow applications to adjust the watering and nutrition of plants according to their type. How does the precision agriculture affect the environment? Precision farming uses the resources only to the extent necessary. Whatever it is needed to that extent only, it uses the resources. So to solve these specific problems, it reduces the losses seen with the traditional agricultural practices and promotes the nutrient balance. So it will avoid excess use and unrequired use of the nutrients so that it maintains the nutrient balance and cut down the nutrient waste of the excess use of the nutrient required by the crops. Also, sustainable agriculture reduces the pesticide resistance development. For example, variable rate technology reduces the dependence of the production on chemicals, which contributes to the long-term productivity of the field. Optimization of transport costs reduces the harmful substance emissions into the atmosphere. Thus, precision sustainable agriculture is the most advanced farming system that allows solving both production and socially significant, including global problems. Precision agriculture solutions like USDA crop monitoring enable everyone to solve these problems regardless of the production scale. Then coming to the soil test crop response, this is a very important aspect. So the nowadays the soil test is required. It is very important aspect nowadays. The precision, like precision agriculture, in the precision agriculture, there is one aspect of the soil test based fertilizer recommendation is also one of the aspects. It is very crucial. Soil test based fertilizer recommendation is very crucial and for optimizing the crop agricultural productivity and ensuring the environmental sustainability. So for accurate nutrient management and for improving the crop yields and to cut down the cost of the on the inputs and to protect the environmental pollution and to maintain the soil health. So there is a need to apply the fertilizers on the basis of the soil testing. So what are the objectives of the soil testing? The main objectives of the soil testing are to develop the fertility status of a given field. What is actually that field particular status, whether it is nitrogen, phosphorus, potash, whether it is low, medium, high, and what is the status of the soil, whether it is acidic or salinic and alcoholic, saline, alkaline, like that, to know the fertility status and other characters in the field. The soil testing is required. And based on the data, then grouping of the soils into different classes based on the relative levels of nutrients for suggesting the fertilizer recommendations or practices. It helps to it helps to evaluate the soil productivity. So based on the soil fertility, the productivity depends. So it evaluates the soil productivity. To identify the soil problems, like just like I told you, problems like alkali, salinity, and acidity, which limits the crop growth. And it is also helpful to predict the probability of obtaining profitable response to the fertilizers. Another objective is to provide a basis of recommendations on the amount of fertilizers to apply. How much we have to fertilizer those we have to apply. That is depends upon the soil nutrient status. So to know the soil nutrient status, the soil testing is required. So to evaluate the fertility status of the soil on a country, soil area and statewide basis by the use of soil test summaries. So this is the importance of the correct sampling. The correct sampling is most important because the taking sampling is very important because the soil testing results, testing of the soil and the results, it mainly depends upon the soil sampling. Soil sampling is very important. If you are not able to take proper soil sampling, whatever the results you are getting are useless. You cannot depend on that useless soil data, which will not be useful unless we take the soil sample correctly. Because the soil in for hectare, 1 hectare soil, it contains 2.24 into 10 to the power of 6 kgs of the soil. But whatever the soil samples, what happens while taking the sample, we'll divide the entire field into, randomly we'll select the different spots in the field, depend upon slope, color, and other vegetation or there are anything else different character. We'll select the different spots and randomly. From that randomized selected spots, we'll take the soil sampling. So if and we have to dig the sample for the 0 to 15 centimeters, we have to take the soil samples as a layer wise from top to 15 centimeters. So like that layers, that layer that also we have to collect from the side where the side is exposed to sun. So like that different spots we'll collect different layers of samples and we'll mix the samples, we'll make the composite sample and we divide the samples into quarterly 4 quarters and we'll reject opposite quarters. Like that, two other opposite quarters will mix up. Like that, we'll repeat till we get finally 1 kg of the soil sample. This is the representative soil sample. For example, if we do not select randomly good spots and we do not take correct soil sampling, so it may not be representative sample to represent the whole soil of the 2.24 into 10 to the power of 6 kg. It will represent wrongly if you do not take proper sampling. So the proper soil sampling is very much important in concluding the soil testing data and on that basis recommending the fertilizers. What are the steps in the soil testing? Collecting the soil samples and preparation is the first step, and extraction and determination of the available nutrients, uh, calibrating and interpreting the analytical results, finally the making the fertilizer recommendation. First one is the collection, proper selection, and then preparation. Then next is the which method we will use, what are the chemicals we will use, how we will extract and how we will chemically analyze the nutrient quantities in the soil. Then whatever the data we get from the analysis, calibrating and interpreting the analytical results, whether it is low, medium, high, and based on that, finally making the fertilizer recommendations of respective crops. So, what are the prerequisites for the successful soil testing service? Effective educational activities, uniform reporting should be there, improved sampling procedures, improved analytical procedures, better correlation and calibration of the data, consistent interpretation and recommendation of the fertilizers, timely delivery of the results are also very important. If we deliver the results after, sowing the crop or after transplanting the crops and after applied the fertilizers, it is no use. So timely, before sowing of the crop, before taking up the crops, if we provide the soil test results, then farmer can take up whatever the apply, whatever the new, fertilizers required, that only he can apply and get the good crop production. So available nutrients. Plants draw their nutrients from air, water, and soil. So the available nutrients like what plant requires: nitrogen, phosphorus, potash, uh, and secondary nutrients like calcium, magnesium, sulfur, and micronutrients like zinc, copper, iron, and manganese, boron, molybdenum. So plants draw these nutrients from air, water, and soil. The bulk of mineral nutrients comes from the soil. So majority, most the bulk quantity comes from the soil only. So soil available form of nutrient is that fraction which is distributed in different discrete chemical forms, which often exists in the state of dynamic equilibrium and constitute the pool from which plants draw. Soil available form of nutrient is also that fraction whose variation in amount is responsible for a significant changes in growth and responses. There are the different methods and of the analysis of the nutrients, and there are the different types of extractants used for the estimation of the available nutrients. For example, available phosphorus, we use zero point 0.5 molar sodium bicarbonate with pH 8.5. This is called Olsen extractant. Most majority of the oil soil testing laboratories it is recommended. For example, suppose soils are acidic in nature, we will use this chemical, ammonium chloride, that is brace extractant we use. It is recommended for soil testing. When the soil is acidic, we use this one. Otherwise, we will use the Olsen extractant for phosphorus analysis. For available potash, we use the neutral normal ammonium acetate, that is one normal. And for available sulfur, we will use the 0.15% calcium chloride. For available zinc, copper, iron, and manganese like micronutrients, we use the DTP extractant, that is 0.005 molar DTPA. We adjusted pH to 7.3. This is diethylene triamine pentacetate DTPA. And gypsum requirement, we use the Schoonover method. Lime requirement, we use the Shoemaker method. So these are the different methods we use for the estimation of the nutrients. Then calibration comes. The greatest challenge in soil testing program is calibration of the test. It is essential that results of soil tests be calibrated against crop responses from applications of the plant nutrients in question. For example, calibration, how to do? It is the process of determining the relationship between the crops and soils. That is correlation of the soil test values with the crop response. If that much crop value, soil test value is there, what is the crop response? If soil test value is increased, what is the crop response? So depending upon that, the calibration has to be done. From the calibrated soil test values, it is possible to predict the extra yield that will be obtained from the addition of the extra amount of fertilizer and the expected yield at the fertility status of the soil. It will also predict the amount of fertilizer to be added to obtain an optimum yield. Soil test values should be calibrated in each soil and for each crop separately. Lack of calibration of such soil test values in one of the most important reasons as to why the soil testing is not so popular. So there are the two different methods of the soil test calibrations. That is, one is the soil analysis correlation approach. Another is the critical soil test level approach. Here the percent yield increase. How we will calculate this? Crop yield with adequate nutrients minus yield obtained in the control plot divided by the crop yield with adequate nutrients into 100. So how will this calibration and interpretation? Here, the most common method in to plot the soil test values. Here, the soil test values in one axis and yield responses on the other axis. So, to plot the soil test values against the percentage yield and to calculate the correlation coefficient between the soil test values and percentage yield response. For example, if these soil test values are increasing, the yield response is decreasing. So at the lower values of the soil test, the to response to the applied fertilizers is the more. So the percentage response, the yield response is more at low soil test values. The soil test value is high means the percentage response to the applied fertilizer is low. So the yield response to fertilizer in relation to soil test value, this is the diagram depicting interpretation you see here. For example, here the levels of soil fertility and profitability and probability of the profitable increase. Here you see the very low fertility, the profitability percentage, probability of the profitability is increasing. When coming to the very high, I mean soil test value, the profitable probability of the profitable increase is less. So lower the soil test value is the more profits to the applied fertilizers, and the less high the soil test value is the requirement, the response is less and profitable to the applied fertilizers is less. So based on the contents of the available nutrient, soil test values NPK, the soils are grouped into classes as low, medium and high. In general, the greatest response can be obtained from the low class, very low class, and the least response from the high class in soil test values. So this is the soil test interpretation. If you see the observe this diagram, here the soil test values. See the here this side very high, optimum, low, very low. This is the nutrient available from the soil. So here the nutrients required, fertilizers required, how much is required, probability of the yield increase. For example, soil status of the nutrients is very high, the probability of the yield increase is one less than 5%. If the soil test values are high, then probability of the yield increase is 5 to 30%. When test value is optimum, the probability of yield increase is 30 to 60%. If soil test value is low, the probability is 16 to 90%. If soil test value is very low, there is a still steep increase in profitability, that is more than 90%. Yield increase will be there. So depend upon the soil test values, the profitability to the applied fertilizers or applied nutrients is more. Fertilizers used at high soil test values are for starter or maintenance purpose. For wherever the soil test values are higher or there, actually there is no need to apply the fertilizer. But to this as a starter, we have to use a little quantity of the fertilizer, even though at the highest soil test values. This is the interpretation, that is another way of predicting. This is the fertilizer nutrient applied, the percentage of yield. The interpretation of the soil test values involves the determining the how much of a particular nutrient will be needed throughout the growing season to provide sufficient supply of this element to the plant for a predicted yield. The lower the soil test value, lower the soil test value for a particular nutrient, the higher is the response to the fertilizer nutrient. High soil test values where probability of increased response to the additional fertilizer is small, 10% only. Medium value soil test values where probability of response to the additional fertilizer is moderate. So like that I have shown in the earlier diagram. Then rating of the soil test values. For example, the next step is we analyze the soils with the different nutrients and different characters. So then how to classify those soils, whether it is having sufficient status of the nutrients, whether it is good or bad, whether it is having low, medium, like that. There are the different ratings of the soil test values. For example, organic carbon is very important for the organic matter content in the soil. So if the organic carbon is less than 0.5%, the soil is called low in organic carbon, and it is when organic carbon percent value is 0.5 to 0.75%, it is a medium, and if it is more than 0.75, it is high. Generally, the most of the soils will be having very that low to medium organic carbon in our Indian soils. Wherever the organic carbon is very low, there is a need to apply the organic manures in at least in required quantities wherever it is available in that particular region. Available nitrogen coming to available nitrogen, if it is less than 280 kg per hectare, it is soil is called low in nitrogen, available nitrogen. If it ranges 280 to 560 kg per hectare, the soil is called medium in available nitrogen. If it is having more than 560 kg of available nitrogen per hectare, the soil is called high in available nitrogen. Similarly, available phosphorus, that is P2O5 content. This is phosphorus content. If it is less than 10, it is not P2O5. It is in terms of phosphorus. If it is less than 10, it is called low. If soils having less than it is in the range of 10 to 25 kg per hectare of available phosphorus, it is called medium. If it is having more than 25 kg phosphorus per hectare, it is called high. Similarly, for available potash, if less than 120, it is low. 120 to 120, 180, it is medium. More than 280 kg per hectare, it is called high. Available sulfur, if it is less than 10 ppm, it is called deficient. If it is more than 10, it is sufficient. So there are for the similarly for all micronutrients, there are the different critical levels. Here we use critical levels and it varies again from soil to soil. In soils, one type of one I mean, critical levels are there. For black soils, it is different. For example, if you take zinc, for red soils, 0.65 ppm is the critical level. For black soils, it is the zero point 0.7 critical level. If it is soil having less than 0.65 of zinc ppm, then we have to apply the zinc fertilizer. If it is having more than 0.65 ppm zinc, we need not apply zinc. Like that, it is the critical level. So critical level is the level below which if soil is having, then we have to apply that particular micronutrient fertilizer. So when copper, if red soils it is less than zero point 0.2, it is deficient. In black soils, less than zero point 0.3, it is deficient. If it is more than zero point 0.2, it's sufficient. Like that, iron less than 3 is deficient in red soils, less than 4 deficient in black soils, manganese less than 4 deficient in red soils, less than 4 in deficient in black soils. Similarly, boron, if it is less than 0.52, the soils are called deficient in boron. If soils are having less than 0.2 ppm molybdenum, it is called deficient in the molybdenum. Critical nutrient ranges, not only the deficiency and sufficiency ranges for micronutrients. We have conducted several experiments in the All India Coordinated Research Project in Micronutrients in Hyderabad Center, where I was the uh, principal scientist and head of that scheme over the 2 decades. We have conducted several experiments. We have finalized the different categories. Under very when zinc is less than zero point 0.3 ppm, we call the soil, it is very low, and zero point 0.3 to 0.75 low, and zero point 0.6 to 1.5 marginal, and more than 1.5 adequate. What happened in general earlier we called if the soil is having less than zero point 0.6 ppm, we were calling the soil deficient in zinc. But it may be okay, points less than zero point 0.6 is deficient, but to what extent? It is very low deficient or very low deficient, like that. Similar categories to define. We have decided these subcategories under the deficient categories. That is low and very low. Similarly, copper less than zero point 0.1, it is very low. Zero point 0.1 to zero point 0.2, it is low. Zero point 0.2 to zero point 0.4 marginal. More than zero point 0.4, it is adequate. So similarly for iron, it is less than 0.25 very low, 2.5 to 4.5 low, 4.5 to 7.5 marginal, more than 75, 7.5 it is adequate. And similarly for manganese, if it is less than 1 ppm, it is very low, 1 to 2 ppm low, 2 to 4 ppm marginal, more than 4 ppm it is adequate. So boron, nowadays the boron and molybdenum are newly emerging micronutrients deficiency. So boron, if it is less than 0.25, it is very low. It is 0.25 to 5, it is low. 0.5 to 0.75, it is marginal. More than 0.75 ppm, it is adequate. Molybdenum, it is less than 0.05 ppm, it is very low. 0.05 to zero point 0.1, it is low. Zero point 0.1 to zero point 0.2 ppm marginal. And if soil is having more than 0.2 ppm of molybdenum, it is called adequate. So then critical levels, the percent yield versus soil test level. groups who already probability of response to the applied fertilizer is small, probability of the response to the added fertilizer is large. Where what happened now, we have to calculate the percentage yield values obtained to a wide range in locations. For example, percent, what is the how we will calculate the percent yield? The crop yields with adequate nutrients, whatever the crop yields will obtain with the adequate nutrients we applied, that and minus yield in the control plant where we do not apply the fertilizers, and divided by the crop yields with adequate nutrients into 100, that is percent yield. Next, soil test values obtained in set plant will generate a single percent yield and one soil test value for each location. Coming to step 3, scatter diagram, we have to prepare, that is percent yield in y-axis versus soil test value in x-axis should plot range in, that is y-axis is the 0 to 100%. Finally, the step D is the overlay. Overlay move to the point where data in the plus quadrants are at maximum and point where vertical line crosses the x-axis, that is the critical soil test level, will define and will arrive. So critical soil test approach, the critical soil test level concept, the first it was developed by the Kate and Nelson during the year 1965. So this is nothing but the level of nutrients below which reasonably satisfactory economic response should be expected from the application of that particular nutrient, and above which the probability of such response is very low. In order to apply this concept, the soil samples are to be collected from each field or to be analyzed. Field experiments are to be conducted with application of the graded doses of the nutrients and response curve is fitted. A scattered diagram of percent yield on y-axis and the soil test values on the x-axis is then platted. It is divided into 4 quadrants, positioning the lines in such a way that the number of points in upper right and lower left quadrants is maximum. The point where the vertical line crosses the y-axis is defined as the critical soil test value. This is the procedure how we arrive the critical soil test value for that particular nutrient. Then establishing the critical level in the soil, the soils collected in each field are analyzed. Field experiments are to be conducted with the application of the graded dose of fertilizers. Response curves are fitted. Scatter diagram of the is to be prepared. It divided into 4 quadrants. This is 1, 2, 3, 4. 4 quadrants. So wherever, so it's the number of points where we have to dissect the lines where the, so that way two opposite quadrants will be having the more number of the plotted points, plotted points. Here, for example, here the maximum, here the maximum. So it is the dissection point. So this point is called, wherever it is dissecting the this A axis, this is called critical level. The point where the vertical line parallel to the y-axis crosses the x-axis, it is defined as the critical level soil test value, critical soil test value. Critical nutrient levels in soils, it varies again depend upon the extractants we use. For example, zinc, if we use the DTP extract, there will be one critical level. For that, whatever the critical level we establish at 0.6 ppm for red soils and 0.7 ppm for black soils for zinc, it is with the extractant, DTP extractant. But when we use then male is number 1 extract, that will be the critical level 0.5 to 3. When we use the HCl extractant, the critical level is 2 to 10. When you use the DTP and the ABDT, that different. So the critical level, it varies. The critical level varies. For example, male is 1.1, DTP 0.6, ABDTP 1.5. It varies depend upon the extractants we use for that particular nutrient. For example, copper. Copper generally for DTPA, as I already told in the earlier slide, at 0.2 ppm. But when we use the malish extractant, the critical level we should be 3. When you use the ABDTPA, the critical level is 1.8. So it varies depend upon the extractants used. So boron, for example, hot water soluble extractants we use. This is the only extractants we are using 0.5 to the ppm is the critical level. For molybdenum, we are using only one extractant, that is the 0.2 ppm critical level. So relationship between the soil and extractable phosphorus. Krishnamurthy and his co-workers during 1963, they worked out empirical relationship between the nature of the soil and the extractable phosphorus. They found that the capacity to fix the added phosphates is largely a function of the type and amount of the clay present in the soil. So that is depends mainly the percentage of the clay present in the soil. So in case of the block cotton soils containing the montmorillonite and type of the clay minerals, the percentage extractable phosphorus will be the 100 minus clay percentage. In case of the red and alluvial soils where the illite clay mineral is dominated, there the percentage extractable phosphorus will be 100 minus 2 into clay percentage. It varies depend upon the clay percentage. Critical nutrient range. Critical nutrient level is that particular figure below which is called deficient. That is critical nutrient value. But wherever critical nutrient range means it is a range from one value, that is a range from X to Y, A to B. Some range is there. So within that range, the crop will it is okay for with that nutrient. If it falls below that range, it shows the deficiency. So instead of critical nutrient level, the critical nutrient range also nowadays we are using. For some of the crops, the critical nutrient concentration is maximum rather than in a minimum. Consequently, it is a more realistic to use the critical nutrient range, defined as the range of nutrient concentration at a specified growth stage above which the crop is amply supplied and below which the crop is deficient. So there are some indicator plants which shows the deficiency symptoms. For example, to find out the nitrogen, the cauliflower and cabbage they were very much sensitive. Whenever the nitrogen is deficiency, they will show immediate deficiency symptoms. Cauliflower and cabbage are the indicator plants for nitrogen, and rapeseed is for phosphorus. Potato is the indicator plant for potash. Potato is indicator plant for potash. Cauliflower and cabbage also indicator plant for calcium. Potato also indicator plant for magnesium. Cauliflower, cabbage, and potato for iron. Sugar beet is for sodium. Sugar beet, oats, and potato for manganese, and sunflower for boron. These are the indicator plants. Even for zinc, it is not mentioned. It is missing. For zinc, maize is the crop which is the indicator plant for the zinc. Soil test based fertilizer recommendations, that is fertility gradient approach. This was put forth by Ramurthy during 1971. The underlying principle is to get as large variation as possible in the soil fertility levels in one and the same field in a given locality. Within the same locality, within the same plant, we have to create the different gradients of the soil fertility. So to elaborate the elaborate field experiment is to be conducted in that field so that the real relationship between the yield and the level of soil fertility could be evaluated without interference from other factors affecting the yield. A large field of certain amount of natural variation in fertility is chosen. It is divided into 4 strips, which are treated with different doses of NPK. The 4 strips we will divide. One strip is with control plant. There is N0, P0, K0. No application of any NPK fertilizer. That is control plant. Another second plant will be off dose, 50% of the recommended dose of the NPK fertilizers. Third one is the normal dose, that is the recommended dose of the NPK fertilizers. And for last one, fourth is the double dose, that is the double the dose of the recommended dose, that is N double P double K double of NPK fertilizers. That are usually applied to the high yielding variety of the crops. So like that, we have to divide the 4 strips and we use the 4 different treatments. So exhaustive crop like sorghum, maize, wheat, or rice are to be grown in the field to deplete the soil fertility in the strips treated with fertilizer. So all these treated, even 4 divided plots for the 4 treatment applied plots, we have to grow the exhaustive crop, which should use the maximum amount of fertilizers, amount of nutrients from the soil, which should exhaust the nutrients from the soil. The type of crops we will raise in this 4 steps. So an experiment is then laid out on this field for the purpose of soil crop response correlation with treatments having different NPK randomly arranged in each strip in such a way that there is a untreated check plot on either side of the treated plant. The soil test crop response work is then carried out taking the yield of the treatments and the average of the 2 control plots into consideration. This approach takes into account the soil contribution and yield level for recommending the fertilizer dose for a particular crop. It will take both soil contribution and yield level. This approach is also called as a rationalized fertilizer prescription or prescription-based fertilizer recommendations. It is specific to a given type of soil and specific to crop and specific to climate situation. The requirement of nutrients is different for the different crops and for different regions. So the efficiency of soil available nutrients and those added through fertilizer is also different from different for different types of soil under a particular set of climatic conditions. So it varies from zone to zone, and it varies from soil to soil, crop to crop. So there is a lot of variation for the soil test crop response approach and soil test based fertilizer equations, developing equations. So therefore, the three basic parameters, that is NR, CS percent, CF. CS percent means that is a contribution from the soil. CF means contribution from the fertilizer. So these are the to be worked out for the specific crop and yield of the development of the prescription-based fertilizer recommendations. That is, NR is nutrient requirement, CS is contribution from soil, CF is contribution from fertilizers. These three are to be worked out. So what is the calculation nutrient requirement? That is kgs per quintals we have to arrive. So total uptake of the nutrients, that is kg per hectare, divided by the yield of grain quintals per hectare. That will get the nutrient requirement in kgs per quintal. And the percentage contribution from the soil. So total uptake of the nutrient in control plots, that is kg per hectare. Available soil test values of the control plots, that is kg per hectare into 100. That will get the percentage of contribution from the soil. Then like that percentage contribution from the fertilizer, we have to calculate like this. Total uptake of the nutrient in the treated plots, that is kg per hectare, minus available soil test values of treated plots, that is kg per hectare, into percentage recovery from the soil divided by fertilizer dose applied kg per hectare into 100. So the percentage contribution from soil, this is the CS means nothing but percentage contribution for soil, CF is the percentage contribution. So like this, the three factors we have to calculate. Then targeted yield equation, how we will develop. Using this basic data, fertilizer dose required for specific yield target is to be calculated by different. This F is nothing but that is NR into T minus CS divided by 100 into S into 100 by CF. Here NR is the nutrient requirement in the kgs per quintal. Nutrient requirement in the kgs per quintal. This F is nothing but the targeted equation. That is. Then F is nothing but the fertilizer required for the kg per hectare. Fertilizer required for the kg per hectare. NR is the nutrient requirement in kg per hectare. T is the targeted yield. How much yield you are targeting to get for that particular crop. Suppose normal yield is 20 quintals. So you want to get 30 quintals hectare. 30 quintals for yield per hectare. Then what will be the nutrients applied? That is the developing the soil test based soil test targeted yield equation. T is the targeted yield. CS is the contribution from the soil, and S is the soil test value, actual soil test value which we analyze in that particular plot, and CF is the contribution from the fertilizer, contribution from the fertilizer. These all values we have to substitute, we will get the that is F, that is nothing but fertilizer required for the kgs per hectare. After calculating these three basic parameters from the yield and uptake data from the well-conducted test crop experiment, these basic parameters in turn are transformed into workable fertilizer adjust equations as below. Fertilizer nutrient dose kg per hectare is equal to that is nutrient requirement divided by the contribution from the fertilizer into 100 into targeted yield minus the contribution from the soil percentage divided by contribution from the fertilizer percent into soil test value. So this is the fertilizer nutrient dose for the particular nutrients. Like this for NPK we have to arrive. So the equations for the nitrogen and phosphorus and potassium are like this. Fertilizer nutrient is equal to that is in X, T and Y. That is the X is the soil test value and targeted equation, and this is this soil test, other soil test value. So like that, all these here this the SN is the soil nitrogen, SN is the soil nitrogen kg for hectare. SP is the soil phosphorus kg for hectare. SK is the soil potash for kgs for hectare. So FN is the fertilizer nitrogen to be applied. FP2 is the fertilizer phosphorus fertilizer we have to apply. FK2 is the potash fertilizer we have to apply. Then the T is the targeted yield. Targeted yield. These are all values we have to substitute and get the final values. Based on this approach, fertilizer recommendations are to be developed for different regions utilizing the different equations. For example, one such equation developed to recommend NPK fertilizers for rice is given below. For example, fertilizer nitrogen is equal to 4.39 minus 4.6723 SN, this is soil nitrogen. So this is the equation developed for the fertilizer nitrogen for rice crop in some particular region. So like that for phosphorus, for like that for potash. So different equations we have developed. We have to substitute the targeted yield value and soil test value. So then we get the fertilizer, fertilizer, that particular fertilizer, which how much quantity we have to apply to that field. It is to be remembered that in the equation, soil phosphorus and potash are considered in the elemental form. For example, from while fertilizer phosphorus and potash are in the oxidative form, that is P2O5 and K2O. Wherever the fertilizers are there, it is in oxidative form, and soil test values will be in the available forms. So for example, rice in black soils of Kurnool and Kadapa of district of Andhra Pradesh, for example, these are the equation derived from the different experiments. Fertilizer nitrogen 6.28 targeted yield 0.40 soil nitrogen. For example, in that Kadapa region, we are going to take up rice crop. We analyze the soil sample. Suppose we get the soil test value, suppose 200 kg per hectare. So the soil nitrogen value 200 kg per hectare means 200 into 0.4 we have to multiply. Targeted yield. Suppose there the average yield is 30 quintals. I want to get the 40 quintals. So we substitute T value with 40 and substitute whatever the nitrogen value you get from that field, you substitute and calculate. So that is the quantity of the fertilizer nutrients we have to apply to that particular plant to get the targeted yield of that crop. So like that, the target that phosphorus and potash also we have to calculate. So some of the equations for the soil test based major nutrients in Andhra Pradesh, combined Andhra Pradesh before dividing. So several the soil test crop response scheme was there, All India Coordinated Research Project. So those project located at Hyderabad Center for Andhra Pradesh, combined Andhra Pradesh, they have developed, they have worked on conducted different experiments on different crops at different agroclimatic regions on different types of soils. They are conducted, for example, rice. For example, rice they conducted in the different zones, coastal region, high altitude region, southern Telangana region, northern Telangana region, like that. Again, different types of soils, sandy soils, clay loamy soils. So different of soils, different agroclimatic conditions, and one crop they have developed the different equations for the crops, different crops. For example, rice for Guntur district for black soils, one equation. And for like that, for other districts, there are some other equation. And for Rabi season, there may be different equation to get the 55 quintals of the hectare. So this equation, we have to substitute the soil test values of that particular nutrient and whatever the target. For example, here we our target is 0.55. The T will multiply with 5. Suppose nitrogen, we got 225 nitrogen, we multiply 225. Then we will get the what is the nitrogen fertilizer we have to apply. So like that, they have developed the different equations for maize in Kharif season in Karimnagar district for sandy soils. They have developed this equation for getting the targeted yield of the 50 quintals per hectare. For sorghum for Kharif season for Miryalaguda for sandy soils they have developed. Sugarcane also they developed for Nizamabad district black soils. Sunflower for Rabi season they have developed for Nizamabad for sandy soils, like that. Different equations were developed for the different crops for the different agroclimatic zones. Basic data for the targeting yield case study. The one example we take one case study, that is Burdwan district of the West Bengal, that is where is the paddy cultivation Ratna variety. These are the NPK values available, nitrogen, phosphorus and potash values. So nutrient requirement, that is kg for quintal grain 0.2, 0.6, 1.53, 3.31. Percentage contribution from the soil available nutrients, that is 36, 33, and 51 NPK. Percentage contribution for the fertilizer nutrients, that is 33, 45, or 58. Then we have to calculate using the values. They have developed the different soil test, depend upon these different soil test values. Fertilizer nitrogen is required, it varies. For example, 140 soil test value, we require 173 kg of fertilizer nitrogen. For 180 soil test value, we require less, 129 kg for nitrogen per hectare. If it is having more soil test value, we require less nitrogen fertilizer per hectare. Depend upon the soil test value. If the lower soil test values, we are requiring more amount of that particular fertilizer. If we are having a higher test values, we are requiring less amount of that particular fertilizer nutrient. So it depends upon the soil test values. So there are some drawbacks of the soil test crop response approach. So these equations are not available for the different crops and different, because there are so many climatic regions, there are so many soil types, and there are so many crops. There are no, equations are not available for the all the crops, for all the regions, for all the types of soils. It will be available for some crops at some agroclimatic zones, for some specific soil types. So development of these involves very cost and very time effective time taking. So these equations are suitable only when available NPK are estimated by the potassium permanganate, Olsen, and ammonium acetate methods respectively. Again, the drawback is the all the equations, the soil testing equations developed with the soil testing values arrived following that particular soil testing methods. For example, soil testing value for potash is when we use the potassium permanganate extractant only, that is valid. For phosphorus, when we use the Olsen method value estimation only, it is valid. And for potash using ammonium acetate extractant only, then that value can be used in that equation. So that are specific to the methods also. If method varies, again the equation varies. This particular developed equation will not be applicable. So the calculation of STCR equation based recommendation, one case study if you take here, recommended fertilizer dose for rice using soil test crop response equation from this data. Let the prescription equations be like that. So, for example, this equation is developed for that for rice crops for that particular agroclimatic region. Fertilizer nitrogen is 4.39 T minus 4.673 SN, that is soil nitrogen. T is the soil test value. Like that, potash, this equation. For phosphorus, this equation. Potash, this equation. Then assume that soil available nitrogen is, for example, you think about that nitrogen is 250 kg per hectare, available potash is 35 kg per hectare, available phosphorus is 35 kg per hectare, available potash is 350 kg per hectare, and targeted yield, for example, you decided 5 tons per hectare for rice. So this 5 tons per hectare we have to substitute it in the place of T, and soil nitrogen, this is the soil nitrogen 250 kg here we have to substitute. And here T 5 tons, 5 tons per hectare. And again SP, we have to use the 35 kg per hectare phosphorus value. Here again for SK, that is potash value, we have to use the 350 kg per hectare. All these values have to be substituted. And here one more is here we have to convert the P2O5 and K2O into P and K respectively, as soil phosphorus and K elemental forms are considered in elemental form in the equation. What happens? The P2O and K2O5 values have to be converted into PK values. For example, soil phosphorus is the 35 into 0.43. For example, soil available phosphorus here is the soil available phosphorus, for example, 35 kg per hectare. We cannot take this value directly here. What happens here? We have to convert into soil phosphorus. 35, that is factor for conversion of the available P, that is P2O5 to P is the 0.43 factors. So we'll get the 15.05 kg of the P per hectare. Like that, potash we have to convert into multiplying 0.83 factor. So then we have to substitute all these values in this equation. So then we will substitute all these values in the equation. We will get 51.43 kg of the fertilizer nitrogen required, 49.55 kg of the fertilizer phosphorus required, and 25 kg of the potash fertilizer is required. So for example, So substituting all these values will get the nitrogen, phosphorus, potash. For example, if the recommended dose of that particular rice crop in that region, that is 52 nitrogen, 50 P2O5 and 0 K2O. When compared with the blanket recommendation, this is what happened here. How much we got? 51 point kg nitrogen per hectare. Fertilizer nitrogen by using the target yield of 5 tons per hectare. Substituting the soil test values, the fertilizer nitrogen is required 51.43 kg. Here that means 52 nitrogen. Then phosphorus 49.55. Roughly it is 50. Here potash minus we got. Minus means it is no requirement of potash. So 0. So the 52 nitrogen, 50 P2O5, 0 K2O is required for that crop in that particular region. Using this equation we have developed. And when it is compared with the blanket recommendation of the 160 kg nitrogen NPK per hectare, lot of fertilizers we are using unnecessarily for getting the 5 tons of the crop. For example, 52 nitrogen is required, but we are using 100 kg. So that means 48 kg nitrogen is waste. And we require 50 kg P2O5 to get the 50 tons for a crop. But we are applying 60 kg. So 10 kg of phosphorus is waste. Here potash, actually we don't require any potash, but we are using 60 kg of the potash. So that that potash using is the waste. So based on the fertilizer soil test based equations and fertilizer approach, soil test crop response approach, we arrive the correct doses of the fertilizer requirement for that particular crop based on the soil test values. So these are some of the references used in preparation of the slides, that is official US government information about the global positioning system and related topics. And mainly I have used one article prepared by the Professor Dr. Peter Kogat, a scientist at EOS Data Analytics, that precision agriculture information I used from this article. And I also wrote some of the articles, that is one article using Internet of Things and machine learning together for agricultural predictive maintenance. That article published by me in the Scopus Journal, and another article I published in the Scopus Journal, predictive modeling of crop yield in precision agriculture using machine learning techniques. The data which I arrived through the experiments and published in Scopus Journals, I utilized some of the data from these articles, and the most of the data from for precision agriculture used from the Peter Kogat data, and the soil test based fertilizer equations and all those things I have utilized in this preparation from the data arrived from the erstwhile Acharya Engineering Agriculture University, and now the Professor Jayashankar Telangana State Agriculture University. Those equations I utilized here to show you, and the critical levels of the micronutrients. That data which was obtained from the All India Coordinated Research Project and Micronutrients at Hyderabad Center, where I was working as a head of the scheme ISHAR project for the 2 decades before my retirement as a director of extension. So these are the references I used for in preparation of this slides preparation. Thank you very much for patient hearing. Thank you.